So you asked me to talk about what psychedelics tell us about the brain, and I'm going to certainly tell you what I think they do. And Peter and Eddie may well disagree, but that'll make it an even more interesting discussion. Well, let's start about things that no one's going to disagree about, which is the pharmacology of these drugs. So when I'm talking about psychedelics today, I'm going to focus totally, through reasons of time, on the serotonergic ones, such as peyote, mescaline, psilocybin, and LSD. And they all are molecular mimics of serotonin. So here's a serotonin molecule, and they all mimic serotonin. There's psilocin, the active ingredient of psilocybin, and there's LSD and mescaline. What they differ in is in terms of their affinity for the receptor. So this is a log scale of the affinity of the receptor, and this is human potency. And this is very high affinity, and this is lower affinity. And you see the drug with the very highest affinity is LSD, and that is very, very potent in humans. And the drug with the lowest affinity is mescaline, which is much less potent in humans. So you need micrograms of LSD, you need milligrams of mescaline. And psilocybin is in between. You use low milligram quantities of psilocybin to have an effect. So the affinity at the receptor is completely predictive of the dose, and that tells us these drugs all work through that receptor. Where are these receptors? Well, they're in the brain, and they're largely in the most recently evolved parts of the brain, parts of the brain which are particularly important in human consciousness. So there's a really high density in thinking and feeling emotional parts, such as the anterior cingulate cortex, the frontal cortex, etc. This is a PET scan using a tracer called SIMB36, which is an N-bomb, in fact, that binds to the receptor, and you can use it to quantify these receptors in living human brains. So this is a real image of a living person showing where these receptors are. But when we drill down into the molecular, the sort of subanatomical structure of the brain, we see that they are peculiarly localized on a layer of neurons in the cortex called the layer 5 pyramidal cells. So let me just give you a very brief overview of how the brain works. And the simplest way of thinking about it is to consider the brain as a series of about 150 billion computers that compute initially in this radial fashion. So each of these neurons is a pyramidal cell. It connects to about 1,000 other neurons and projects to about 1,000 other neurons. So each of these neurons is a little mini computer, and there are about 150 billion of them in the brain. And the primary computing is done in this axis here between this multiple layers of the cortex. But then the next level of computing, which integrates the particular local computing, is done across the brain, and that is done through these layer 5 pyramidal cells here. And these have very high density of the 5-HT2A receptor. And when you take a psychedelic, you stimulate that receptor and you depolarize these cells, and they fire like crazy. In fact, they fire so actively and they're controlled. These are such important neurons that they have their own special set of inhibitory neurons called the GABA interneurons or chandelier cells, which regulate them. So there's a very sophisticated network that has to be kept under control. Otherwise, your brains would always be in the psychedelic state. And these neurons are critical for cross-cortical information transfer. They receive information from other parts of the cortex, and that allows you to integrate cortical function. And then they disperse information to other parts of the cortex. But the other thing is about these receptors is they're also likely to be involved in neuroplasticity. And it's becoming increasingly plausible that neuroplasticity is critical to the enduring impact that psychedelics have on people's understanding of themselves, and also potentially more mundane phenomena such as motor activity. Now, because no one had ever done it before, 10 years ago, we did the first imaging of psychedelics using modern brain technology. This is a technique called arterial spin labeling. We gave healthy volunteers that had had a psychedelic before, but not in a scanner before, psilocybin in a scanner, and we had the expected experiences. They had the experiences of visual hallucinations, alterations in ego, strength and connectivity, or distorted perceptions of time and space, etc. When we did the scans, to our amazement, we didn't see any increase in activity at all, which was what we had predicted. We just saw some decreased activity, particularly in these three regions, the posterior and anterior cingulate and the thalamus. And I showed you on the previous slide, there's no 5-HT2A receptors in the thalamus. So the primary effect, we believe, is through stimulating the inhibitory neurons in these regions. And the thalamus is probably a secondary phenomenon. And that was a remarkable finding. We didn't believe it. We thought it was something that had gone wrong. So we repeated the experiment using a different form of MRI imaging called BOLD. Got the same results. So therefore, we concluded it must be true. And how can we make sense then of the emergence of powerful alterations in experience, and often with profound visual components, by switching off one or two parts of the brain? And to make sense of it, we started by exploring this concept of the connector hubs. And this is one of the earliest diagrams of the human connector, and which bits of the brain connect with which other bits. About the same time as we were doing our work, the idea was developing that there were two major hubs in the brain, the frontal hub and the posterior hub. The posterior hub integrates all your senses, your sense of hearing and seeing and feeling and proprioception, etc. And the frontal part, the frontal hub, integrates your thinking, your feeling, your emotions, and your memory. So these are like the two conductors of the brain's orchestra. And the analogy I like to use is that because psilocybin switched off these centers, the brain was able to break free from its traditional control and behave differently. So if it will be like an orchestra, when you get rid of the conductor, the orchestra might decide it's not going to play Mozart anymore, it's going to play jazz, which of course is one of the ways jazz developed, through the use of cannabis, which also disrupts this network to a lesser extent. Now, what was particularly appealing about that discovery was it accorded very well with the insights that this man had made as a result of his own experience of mescaline. And he wrote about them in The Doors of Perception, this most remarkable book. And the title of that book is taken from this quote by William Blake, the English mystic and painter, the man that wrote the words to the song Jerusalem. And like many great, I suppose probably all great artists, visual artists anyway, Blake could see things differently. And he realized that most people couldn't. And Blake wrote this: if the doors of perception were cleansed, everything would appear to man as it is, infinite. For man has closed himself up till he sees all things through narrow chinks of his cavern. And Huxley, the reason Huxley used that quote, because he realized that mescaline had opened up his doors of perception in the way that Blake perhaps had hoped one day people would discover a method of doing that. And then Huxley said this: he said, the brain is an instrument for focusing the mind. The implication being that mescaline disrupts the brain and therefore takes away the constraints that the brain puts on the mind. And our work has actually proven that he was right, and I'm going to explain that now. So I'm going to use vision as an example. And for those of you who are not neuroscientists, I'll just talk you briefly through the processes whereby you see things. So you might be looking out, if you're fortunate enough to be somewhere where it's sunny, you might be looking out today through a window at a wonderful sunset. But your brain doesn't take a picture of that sunset. The way you understand and appreciate the sunset is because the photons from the sky and the clouds go into your retina. And the retina then turns those photic stimuli into electrical activity and sends a whole range of different electrical signals up the optic nerve into the brain and into different parts of the brain. And the visual cortex occupies or is responsible for about a quarter of the whole of the cortex. And different parts of the visual cortex tell you what you're seeing, how you're seeing it, where it is, what its color is, if it's moving, etc. And that's all done by a reconstruction. So the visual parts of the brain reconstruct from those electrical impulses a prediction of what you have seen. And that is true for all sensory inputs. The brain reconstructs something that it predicts will be out there, based on the best evidence that it has accumulated over decades of being a brain, and also over minutes or hours of knowing what you were doing. But when you disturb the layer 5 pyramidal cells with psychedelics, the brain cannot reconstruct. At times it's not able to, because those neurons are vital for that cross connectivity that allows you to put together the different elements of the visual system. And what is peculiarly interesting about the psychedelic, particularly psychedelic hallucinations, is that because the brain cannot make the image that it is seeing or was expecting to see, you end up experiencing something which you never experienced before in your life, which is just the primary working of the visual cortex. We know that these kind of elemental images and spirals and spiders' webs and hexagons, etc., they are the primary processes of those little computers, those pyramidal cell computers in the primary visual cortex. So under psychedelics, you can, on occasion, see the primary workings of your visual system, because the higher level processing is disordered. And that is, as I say, kind of proof that Huxley was right, that the brain is actually an instrument for focusing the mind. And if you take away that ability, you end up seeing things differently. But by switching off these control centers, something else very, very interesting happens. Not only do you see primary processes, but you also increase connectivity, because the brain is an extremely efficient organ. On the left-hand side, you see normal brain activity, and these are statistical maps. Each of those images has 7,200 connections. But on the left-hand side, effectively under placebo or in the normal state, most of the connections in the brain are around the edge. That's called the small world concept. Visual cortex talks of visual cortex, auditory cortex talks of auditory cortex. Of course, there's got to be some cross-cortical talk, because if you see a snake about to bite you, you need to move your arm. So you need to link visual and motor systems, etc. But most of the work is around the edge. And the reason for that is that that's the most energy efficient way of using the brain's ability. And that the constraints on the brain to make it work in a small world are driven by those centers I showed you in the earlier slide. But under psilocybin and other psychedelics, that constraint breaks down, and so the brain can talk to basically any part of the brain can talk to any other part, and we see massive increases in connectivity, and that is important for two reasons. That is important because it allows people to access things which their brain has actually restrained them from accessing over maybe decades. But it also allows them to put things together in a different way and to come up with solutions and insights and to their problems. And this, I think, is one of the reasons why psychedelics are so powerful in changing the way people think about many things, both themselves and the world. Getting this published was a bit of a challenge. We had referees that were very cynical. One of them said, it's just all blood flow changes from 5-HT receptors on blood vessels, which it isn't, because those receptors are on all blood vessels, and we saw rather localized changes. What do you expect? Another one said, what do you expect if you messed with the brain with psychedelics? Well, I'd expect actually better refereeing, frankly. And this is part of the whole stigma which has been built up over the last 50 years. The denial of the value of psychedelics in an attempt to stop people using them has actually even distorted scientific reason about them. And I want to give you another quote from Huxley, which is particularly pertinent to this. Orthodoxy is the die-hard of the world of thought. It learns not, neither can it forget. And this is a hugely important challenge to science. Orthodox science wouldn't allow us to study these drugs. Having studied them, we've seen that the orthodoxy of how the brain works has been rather disrupted. However, we took the challenge of the referees to prove it wasn't just blood flow. We took the challenge by looking at MEG, magnetoencephalography. This measures electrical activity in the brain through the magnetic fields that the electrical activity produces. And we found the same thing. We found that these two areas, the anterior and posterior cingulate regions of the brain, were dramatically perturbed by psilocybin and also by LSD. And that perturbation is demonstrated here by disruption of power in these different frequencies. The brain, as you probably know, is an oscillatory machine as well as a series of neuronal connections. And those oscillations are critical for brain function. And the oscillations occur at different frequencies. And these are decreases in the power of the oscillations, low frequency and high frequency. And this is particularly important image here because the high frequency oscillations that we see down here are the oscillations which underpin much of the sort of current understanding of how neurons interact and new information is laid down. So it is a real effect, it's not a blood flow effect. And if we look at MEG data we've generated with other drugs, we see that the psychedelics are very, very different from any other drug. So here's psilocybin, here's LSD. Blue means reduced power of oscillatory frequency, and red means increased power of oscillatory frequency. So drugs that put you to sleep or put you into a state of anesthesia, like these drugs, they increase frequency. They make the brain much more ordered, everything fires simultaneously. And that is a very uninteresting state. When you're anesthetized, you don't learn anything. You can't even think. So increased synchronicity is limiting to brain creativity, to brain activity and function, whereas decreased synchronicity, which we call increased entropy, is a state in which your brain can think much more fluidly. And for those of you who are still drinking, then alcohol, unfortunately, is more of a synchronizer than a desynchronizer, which is why you become very boring when you're drunk to your friends. And based on that series of data, I've come up with an idea that there are at least two separate dimensions of consciousness that we can understand in pharmacological terms. There's the normal awaking consciousness, which we're all using today, which is driven by the transmitter glutamate, an excitatory transmitter, and moderated by the transmitter GABA. And glutamate activity is critical for arousal, for attention, for memory, and what I call parcellation, the laying down of the precise and amounts of information that you learn on a basis. The amount of information you've already learned from my talk is enormous in terms of the definition of the visual and auditory inputs you've achieved and the memories you've laid down. And this process of very beautiful, precise, discrete parcellation is driven by glutamate. But if you stimulate the 5-HT2A receptors, you get a different kind of consciousness. It doesn't affect memory. People can still tell you about the trips they had 30, 40, 50 years ago. But it does alter, in some ways, the valence of them, so the affect of them, the meaning of them. And the reason for that, I believe, is that the psychedelic state is a state of greater integration. It's the bringing together of insights and experiences and maybe percepts from different parts of the cortex and subcortical regions in a way which is not normally allowed. So this is the very energy efficient. This is the way the brain does its business generally. But under psychedelics, it can do it slightly differently. And that can be very helpful. And then just briefly, I want to explain why we went from what we had discovered with psychedelics to treating depression. And we did it because depression is the #1 cause of disability in the Western world. Treatments fail in about 1/3 of people, and it's becoming more and more prevalent in young people. And there were two reasons we moved into depression from our psilocybin experiments. The first was that psilocybin attenuates brain activity in this region called the subgenual cingulate cortex. And this area of the brain is intimately linked with depression. And pretty much all known treatments of depression attenuate activity in this region as people get better using these different treatments. So that was the first reason. Psilocybin might be antidepressant because it does what other antidepressants do. And the second reason was because the areas of the brain which are profoundly disrupted, those control centers, are what's called the default mode network. And here you see the default mode network under placebo. It's the anterior and posterior cingulate and these two lateral parietal regions. And this is where your sense of self is, where your ego is embedded. And psychedelics disrupt that. It disappears under psilocybin and under LSD. And that is why people have ego dissolution, because that's where your ego is. But the default mode network is particularly interesting because it's overactive in depression. Depressed people have more of their brain engaged in this default mode network. And the default mode network you measure when you're not doing things, or when you're actually just lying quietly in the scanner, reflecting about yourself. That's the default mode network. And there's more of the brain is in the default mode network in depressed people than in controls. This overactivity is driven by this region, the subgenual cingulate, which is overactive. And here you see there's a strong correlation between rumination and the default mode network. The more the default mode is engaged, the greater your rumination. And that's, of course, one of the key features of depression. Depression is a disorder with intense introspection. It's an internalizing disorder. People think repeatedly about themselves, errors they've made, mistakes they've made, guilty thoughts they have. And that's all driven out of the default mode network. And we thought, well, if we can disrupt the default mode network, maybe we can disrupt depressive thinking. So eventually, after years of struggle, we managed to do the study, and we were right. We produced profound benefits after just a single trip in people who'd failed on conventional treatments, both drugs and CBT. And I believe many of you would have heard one of those patients talking this morning. So this was an example of what you would call translational science. You do a study, a brain study, you find an effect that has potential for a treatment, and then you explore the treatment. And this is truly, I think, one of the most interesting things I've ever done, because I didn't start out thinking I would use psilocybin to treat depression. I assumed we were just going to use it to explore the psychedelic state, but it turned out to have clinical utility as well. And one of the interesting things that the patients who've been through this experience talk about is a kind of brain resetting. And I'll just give you these couple of quotes before I finish. So here's one of our patients saying what the effect of psilocybin on his brain was like when you defrag the hard drive on your computer. I experienced blocks going into place, things being rearranged in my mind. I visualized as it was all put in order, a beautiful experience with those gold blocks going into black drawers that would illuminate. And I thought, my brain is being defragged. How brilliant is that? And this is sense that patients have, that their brain is not under their control when they're depressed, that it's doing things that they don't particularly want to do. And this patient went on to say, my mind works differently now. I ruminate much less, and my thoughts feel ordered, contextualized. Rumination was like thoughts out of context, out of time. Now my thoughts feel like they make sense with context and logical flow. And this is the disrupting of the ongoing habitual negative thinking of depression and making things make more sense. And if you want to read more about it, here's a very simple review we did just a few months ago, which takes you from the single pyramidal cell with its depolarization effect of psychedelics through the changes in brain entropy to the connectivity, and it helps make sense of those human consciousness changes that psychedelics produce. So thank you very much, and I'll hand over to the next speaker, and we'll take some questions in a minute.